Okay, so it's like, I'm wearing it. But I figure every time I wear a suit for something, now I'm wearing it. And then you get the same thing for ASM. Well, I got my ASM thing. So I figured, it's in honor of those guys, I wear my outfit.
Vince, do you have my slides?
Yes, I do.
Oh, okay, good. Put a preemptive for a couple of minutes.
I heard that's great. Did you want me to?
I'm just talking about ASM awards.
I'll go a couple slides and then I'll move to. Okay, good. So we're gonna get started. Welcome to the 802nd EPAASM monthly meeting. So today we're very excited to have Dr. Kim Orth from UT Southwestern. She's a professor and HMI investigator to give a lecture. She is also an ASM distinguished lecturer. And the ASM DL program has been around for more than 55 years and delivering more than 600 lectures. And this is a mechanism for National ASM to deliver exciting scientific lectures to ASM branches at a local level. And it's funded by ASM and the Wixman Foundation for Microbiology. And so this is a smooth transition to getting you to join and renew National ASM membership. This is a way to really nurture our community, to establish the networking with your colleagues around the world, to advance your career and your science. And so please go to asm.org/membership to join. It begins October 1st, but it really starts in the new year. So you still have time. And so now Irv is going to talk about the ASM awards and prize for National ASM.
So
You come over here.
Can I just, which one do I?
To
move along
yeah.
this one. Okay. Hi, everybody. I'm Irvin Shampton. I'm the chair of the ASM Subcommittee on Awards. Many of you may know about the national awards that are given each year by ASM. These are really important awards, and they really recognize excellence amongst the members of ASM. And I just wanted to kind of urge you to consider nominating folks from our region, people that that may be deserving for a national award. And I just wanted to tell you that the award nomination window opens up on May 1st and will go through June 26th. And we have kind of several buckets of awards. One, we have a large portfolio of research-based awards. Some of you may be familiar with some of them. Some may be new to you, but they really cover a whole spectrum of disciplines across ASM. So we have the research-focused awards. We have 2 awards in the education space. We have an education committee. I think people, at least in our branch, know of people who are involved in developing educational programs, not just at their institution, but have impact outside their institution. And these would be appropriate awards to nominate folks. We have a group of clinically focused awards. So these are people that are in the clinical space, clinical microbiology. And I want to mention that we have a new award this year for early career clinical microbiology research. So we have some early career awards for some of the other areas of the awards. And so this is a new one in clinical microbiology. And then finally, we have a whole bunch of what I would call service-focused awards. Obviously, an award for service, and that's a broad spectrum of service to the society, the microbiological sciences. And all I would say is that you don't have to have impact just within ASM. It can be impact anywhere outside of ASM, other societies that are involved in the microbiological sciences. So go to asm.org/awards, and you can see the descriptions for all the awards. You can contact me if you have any questions. I should say we have 6 selection committees. Our subcommittee does not select anybody. We oversee the whole process. But if you have a question about appropriateness or want clarification about the type of award, contact me. I'd be happy to talk to you. All right, thank you.
Thank you, Irv. And so just to reiterate, right? So please join and renew your membership at ASM. And on the same idea, please join and renew your membership for EPAASM branch membership, right? So we had our 100th year anniversary a couple of years ago. We established in 1920, but we wouldn't be here without the support of our members, our very active student chapters, our education committee, and our executive committee. So we have, of course, these monthly meetings, but we also have the annual clinical microbiology symposium, as well as our Philadelphia Infection Immunity Forum. And our education committee is always doing scientific outreach and education programs. And recently they've gone to various science fairs in the area. And these are some of the special awards that EPAASM gave out. The top winner was using bacteriophage to combat potato black leg disease. And other honorable mentions, including looking at antibiotic efficacy used with antibiotics with gold nanoparticles, and also using a fungal biofilter to reduce nitrogen pollution. So this is really our way to help the next generation of scientists from high school, right? All right, so just go to our website at epaasm.org, and you can join both national membership as well as for faculty and postdoc is $20 per year. And for student, it's free. So all the food and drinks outside is free for you. Just join the membership. And here are some of the remaining events for the year. So next in May 22nd, we have our student chapter organized a monthly meeting, which we have featured 3 great student talks. Those are the winners from our Philadelphia Infection Immunity Forum poster winners. And we have our keynote speaker, Megan Povalones from Villanova University. And then, of course, we have the ASM microbes in June. And then on June 19th, we have our last Norman Willett Memorial Lectures organized by the Education Committee. And Stacy Latini will give a talk on education. So without further ado, I'll give Jason to introduce our speaker for the night. So I'm going to switch the computer. Don't be alarmed if you're on Zoom. So.
All right, thank you. So I'm Jason Heindel, and it's my pleasure to introduce our guest speaker for today, Dr. Kim Orth. She is an American Society of Microbiology distinguished lecturer, and she's very distinguished, so much that I have a list of many wonderful awards that I'm going to just pick a few from, because she's going to have a slide near the end where she's just going to kind of go over her career. And so she is currently an investigator with the Howard Hughes Medical Institute and a professor in the Department of Molecular Biology at the University of Texas Southwestern Medical Center in Dallas. Her undergraduate degrees from Texas A&M, She then started her graduate career at UCLA, getting a master's at UCLA, and eventually moving back to UT Southwestern to get her PhD. She's done several postdocs. I know of at least one, maybe two at Michigan. But what's more interesting now, she's the Earl Forsyth Chair in Biomedical Science, W.W. Caruth Jr. Biomedical Scholar, and a couple other important highlights. In 2016, she was elected to the American Academy of Microbiology. 2022, she was an elected fellow of ASBMB. And in 2020, she was actually elected to become a member of the National Academy of Sciences. So it's very, very distinguished. I think you'll note here in her talk, and those who have met with her, that not only is she an amazing scientist, bacterial pathogenesis being her focus, using bacteria to sort of understand eukaryotic biology as well. She's also a great mentor and very well known for her mentoring and her really supporting the next generation of scholars. And so if you haven't interacted with her yet, I hope you get a chance to, because I think you'll experience that. So please join me in welcoming Dr. Orth. Thank you.
Okay. Everyone hear me? Yeah, good. Thank you for that introduction. That was very kind. And thanks for the invitation. This is, I would encourage, if you have an opportunity in the future to be able to be an ASM lecturer, you should do it. You really get to travel to some really unique places and meet fun people and people that love science all over the country. So it's been, it's been, even with COVID, it's been quite an honor. So I don't want to move anything. So here we go. So, yes, I can talk. Oh, is the mic? Can you hear it now? Is that better? No, is that better? Uh-oh, you don't know. Huh, okay. I put it near my throat. Is that okay? Can you hear what I have in the back? You can hear? Okay, I'll try to talk louder. And let's see if we can use the pointer. Maybe not. It doesn't, that's okay. I'll just, we'll go through it without the pointer. I'll tell you where to look, okay? All right, so I'm gonna tell you about those things, black spot, black death, and black pearl. And because it basically tracks different things that we've worked on in my lab over the last 20 years, or almost 22 years now. And so it's kind of fun. So it's going to be a long story, but it's a fun story. So let's see. It's not moving. Okay. So one of the things that I first want to bring up is the reason that we study these things. And it's because during infection, bacterial pathogens disrupt and deregulate molecular switches. And by uncovering the activity of these bacterial virulence factors, we discover molecular switches and tools. So I want to bring this to a practical aspect. So that means, like, we know about switches in our house. We have binary switches, on-off switches for lights. We have sliders, you know, basically dimmers. We have dimmers in here, it worked. And then we have, in Texas, this is very important in August, we have to have an air conditioner. We have to be able to turn down the thermostat. And we have water, and we can regulate the water, how fast it flows and if it's warm or hot. So imagine one day you come home and your house is so hot. It's so hot, and it's August in Texas. So it's 101 out, it's 101 inside your house. And then you realize every light in the house is on. It's very, very bright. And then you find out that all the water faucets are on, and you can't turn any of these things off. It's just the way the environment is. And basically what happened is, something, we'll call it an alien, came into your house and it changed the environment. It likes it hot, it likes it bright, and it really likes the water running because that's the sound it wants to hear. So you're going to call the repairman, and you look for a very important device that's in the house, and that's the remote control for the TV. Because while you're waiting for the repairman to come, you're going to watch TV. Nope, somebody hid the remote control. So now you can't even change the environment to the way that you like it. And the reason I'm telling you this story is because this is exactly what pathogens do. They come in and they design an environment which is the best for them to be virulent and propagate and move on to their next host. They basically change the circumstances for their favor. And so a good example of this is with Vibrio cholera, when the water and sewage treatment is inadequate, as in post-earthquake Haiti. cholera can spread rapidly. And why is this? Because the pathogen is ingested, it colonizes in the intestine and becomes hypervirulent. It secretes a toxin, induces watery diarrhea, and then it passes on the pathogen to the new host. So it's basically changed the environment so that it can propagate and become virulent. So in my lab, the tools that we like to study are things that are produced by gram-negative bacteria that have this thing called a type 3 secretion system. It's a syringe-like structure. And what the bacteria do is they inject these virulence factors or effector proteins from their cytosol in the bacteria into the host cytosol. And then they go and manipulate the cellular response to infection. So the first story that I'm going to tell you about is a very old story. And it was when I was an assistant professor. I moved from Michigan and I was working on this effector called YOPJ. And we discovered these fun things about YOPJ. I'll tell you in a minute. And this student was one of my first students. I'm going to tell you about my other first student. She was on this project too, but you'll see what happens. And she was very interested in studying this virulence factor. So what is this virulence factor? So it's YOPJ from Yersinia, and Yersinia can cause gastroenteritis. It also can cause the plague. But all of these bacteria have the same virulence plasmid that encodes this type 3 secretion system, and it injects these virulence factors. And one of them was this factor called YOPJ. And it was a 30 kilodalton protein. And at the time, you couldn't tell anything that it looked like. There was no alpha fold or anything like that. And basically, what we knew is it disrupts signaling in cells. And so I went in using a couple of different tools. And what we discovered is that YOPJ can inhibit all the MAP kinase signaling pathways and the NF-kappa B pathway. And so when it did this, what it essentially did is it promoted apoptosis because it prevented protective factors from being made. And when stimulus was given from the infection, then basically there was no protection for the host cell and the host cells would die. So the next thing that we did is we took this sequence and we compared it to we derived its secondary structure, and then we compared it to all the proteins that had been crystallized. And what we found is that YOPJ looked very similar to a cysteine protease. It had a catalytic triad, and it had all of the right positions for its secondary structure. And so we looked at this and we thought, well, if it's a cysteine protease and we mutate the catalytic residues, it will no longer be able to inhibit these signaling pathways. And that's exactly what happened. So now we have a hypothesis. We have a great hypothesis. We hypothesized that YOPJ, and what I didn't tell you, and I'm going to step back for a minute, is many of these effector proteins are shared by many different bacteria because they can transfer DNA between them. And so many species of gram-negative bacteria have YOPJ homologs, and they all have the catalytic triad. And every time you knock out one of those important residues, whether it's in Yersinia or Vibrio or Salmonella, it disrupts the activity of this protein. So we predicted that they were all going to be dependent on these catalytic residues. And furthermore, based on the proteins that it looked like, we thought that it might be a protease and that it might cleave things. Because what YOPJ looked like was a adenoviral protease and another protein called ULP1, which is a ubiquitin-like protein protease. So it can cleave a peptide bond and it can also cleave an isopeptide bond. So what does that mean? So many proteins can be decorated with other proteins like ubiquitin, and that makes an isopeptide bond. But the cleavage site's the same as if you're cleaving a peptide bond or you're cleaving the protein off the side of a protein. So we predicted it had peptidase activity. It would change the molecular weight of a protein. And if it had isopeptidase activity, it would cleave a post-translational modification like ubiquitin or sumo or something like that. So we did this really sophisticated alignment. So we have YOPJ, we have ABP, and we have ULP1, and they all have this catalytic core with this catalytic triad. And so we wanted to figure out what YOPJ was doing. And this is where the second student, I'm going to tell you about her. They both started at the same time, Sahini and Renee. But we couldn't figure out what YOPJ was doing. For about 2.5 years, we did the best controlled protease experiments that anybody could do. And YOPJ had absolutely no activity. We tried all these different substrates and we were quite flustered by this. Sahini was convinced that this was a really important project and she was going to stick with it. But for many of my students, what I do is I give them a hip pocket project. So Renee had this hip pocket project. And now what we're going to talk about is the black spot, because it turns out there's a homologue of YAPJ, not really YAPJ, but of ULP1 in Xanthomonas campestris. And what it does is it causes black spots on tobacco and tomato leaves. And so she decided that she was going to study this protein. And basically what the protein looked like, it looked just like ULP1, and it had all the nice colored boxes. It had its catalytic core and its catalytic triad. And so she went to look at this protein and she isolated it, and she said, sure enough, it is a type 3 secreted protein by this other bacteria, Xanthomonas campestris. And it is a cysteine protease. It looks like a cysteine protease. It has a catalytic triad, but it actually had desumoulating activity. So it actually had protease activity. So this was really nice that she was getting positive results, even though all our YAPJ results were still negative. And then what she figured out is that ZOPD has substrate specificity. So it likes plant sumo, but it doesn't like yeast sumo. But this was a beautiful thesis project. And this protein was so well behaved that she decided that we had a crystal core and she was going to try to do crystallography. And that's what she did. And she solved the structure of ZOPD. And then she took that structure and she overlaid it on a structure that Chris Lima had solved of ULP1 and its sumo substrate, this peptide substrate that it works on. And so she threaded her ZOPD onto the ULP structure and she learned a lot. And one of the most important things she learned was what the substrate looked like and what the protease was recognizing. So now what I'm going to do is go to this really high quality images of these proteins. And I'm going to tell you that that little SB domain, the blue domain, is what recognizes sumo. And remember, we're thinking YAPJ might be recognizing sumo or ubiquitin or net8, or it might be a protease. Unfortunately, YAPJ and its homologues, that VOP-A guy, which I'll talk a little bit about in a minute, it doesn't have that sequence. So there's no way it could recognize that as a substrate. So what that told us was that this missing substrate binding domain was telling us that YAPJ does not interact with ubiquitin, net8, or sumo. So all this hypothesis that we had that YAPJ was working on these substrates was probably not right. And so 3 years later, we're still studying YAPJ and we're asking, well, what's its mechanism? It has what we think a catalytic triad. And is YAPJ a hydrolase? Is it a protease? And I'm telling you, as an assistant professor, I was very frustrated. We had other projects that were working, but the YAPJ project that I was thinking, oh, this is what I have to do. I won't get tenure. What am I going to do? And finally, I was at a meeting. I was complaining, but I didn't tell you something about me at the very beginning. I'm not a microbiologist by training. I'm a biochemist by training, and I study molecules that come out of microbes. And a very influential scientist, Tony Hunter, came up to me when I was complaining. He goes, Kim, you are a biochemist. Use biochemistry to figure out this problem. So Sahini was at the meeting too. So we went back and we decided, okay, we're going to set up an assay and we're going to try to figure out how this works. So the first thing that we did is we decided what we would do is put these MAP kinase signaling pathways and NF-kappa B pathways in a test tube. So we isolated lysate from cells that expressed a vector alone or YAPJ or the catalytically inactive form of this. And then we take those lysates from starved cells because we basically dampened all the signaling of these cells. And then we added something to turn on the signaling. And in this case, what we added was BRAF. And what you can see is that if you look at the downstream of the kinase, remember YAPJ inhibits MEK. So downstream of that is ERK. So we looked at ERK phosphorylation. If YAPJ is present, we inhibit the phosphorylation of ERK. So in those lysates, now we've got the whole thing in a test tube now. So that's really cool. And so then we took those lysates and we checked the NF-kappa B pathway. And sure enough, when we asked James Chen, we said, can we have some TRAF6? And he said, no problem. So he gave us some TRAF6 and we look at phosphorylation of I-kappa B and we see that YAPJ.
Ma tu riesci a seguire?
Can hear. Hi, you have to turn on your speaker. Okay, good. Okay, so So we saw that YAPJ can inhibit the NF-kappa B pathway. So that was very nice. It was a very satisfying result. So remember, we thought YAPJ was a protease. So that was going to shift the molecular weight of something. So we looked at every protein in the NF-kappa B and the MAP kinase signaling pathways. And every time we did a Western blot, we got the exact same result. So this was a problem. So now we had to think of another way of doing this. So we thought, well, what we'll do is we'll recreate this MAP kinase signaling pathway in bacteria because they do not have it. So what we're going to do is we know that YAPJ binds MEK. It binds the kinase that it inhibits. And we know it inhibits its activation, and its activation is done by phosphorylation, and we can follow that. So what we did is we took a vector YAPJ and C to A, and we co-expressed it with this kinase MEK in bacteria. And then we said, maybe it's cutting in a little piece of it, or it's screwing up its dimerization. So we do some biochemistry and we look at these guys, and they all make beautiful dimers. So that's not the problem. They all look the same. But then Sahini did a very clever experiment. She said, well, if YAPJ is around, and we purified YAPJ away from this kinase, so the kinase is very, very pure. And we said, can the kinase be phosphorylated? And we said, how can we check that? Well, we can go back to our in vitro assay and just add it to an active lysate and ask if these recombinant proteins can now be phosphorylated. And what we found is, if we co-express YAPJ with a MEK in bacteria and then isolated that MEK, it can no longer be phosphorylated. Whereas the other two, the one with the vector, the C to A, can be phosphorylated. So there's something different about that kinase that we purified. So then we thought, well, maybe it's cutting a little tiny piece and we just can't see it because this isn't a good enough resolution on the gel. So what we decided to do was determine the total mass of the kinases and try to figure out how they changed. And this was a big surprise. So the kinase expressed with the vector and the kinase expressed with the C to A had the same mass. But when we looked at the kinase expressed with YAPJ, it didn't decrease in molecular weight, it increased in molecular weight, 126 Daltons. What is that? So one other hint we had is we had a very good person that worked on the mass spec, Yanli. This woman is phenomenal with doing post-translational modifications. And she said, you know, Kim, it's not really 126. I said, what is it? She said, it's 42, 3 times. And I went, oh, well, 42, we know what that is. That's acetylation. And acetylation happens on amines. It happens on the N-terminus of a protein, right? And it can happen on a lysine residue. So she said, yeah, but look at the peptide analysis. These are the 2 peptides that are modified by this acetyl group. And it turns out that it can't be on the N-terminus of these peptides because an enzyme cleaved. And there wasn't J around to modify the N-terminus. And if you want to modify the lysine residue up there that's between these 2 peptides, you can't do it because if it's modified, it can't be cleaved. She said, in fact, when I look at the B ions and the Y ions, what it tells me is the serine in the first peptide is acetylated, and the threonine in the second peptide is acetylated. And if you get a long peptide with these 2, the lysine is acetylated. And that makes sense because it can't be cleaved, right? So what is special about these? I didn't believe it for a week. Okay, finally, we say, okay, what is special about these? And so we go to all the kinases that YAPJ can inhibit. And it turns out the serine and threonines are totally conserved. And it also turns out that these residues are the same on the activation loop that are phosphorylated by upstream kinases. So what that told us is that acetylation prevents phosphorylation. And then, of course, this is a hypothesis, so you have to test it, and you test it with C14 acetyl-CoA, which takes forever to see on a gel, but we did it. And what we found is that YAPJ is, in fact, an acetyltransferase, and it modifies serine and threonine residues on these particular kinases to inhibit the signaling pathways. So essentially what happens is normally this triple, this MKK, can phosphorylate the MKK, and it activates it by phosphorylation. But when YAPJ is around, what it does is it acetylates the serine and threonine, and that prevents phosphorylation. So we go to publish this paper, and we do, and we basically say that YAPJ acetylates critical residues on the activation loop and inhibits activation of the kinases by phosphorylation. But there were people, there were reviewers, and they said, what happened to the hydrolysis hypothesis? And I think this is actually probably the coolest part of this whole story. And that is that there's two ways to acetylate a protein. The first way is by a direct transfer mechanism. Both substrates bind, and then the amine in this case is coordinating to be the nucleophile that attacks the acetyl-CoA. It takes the acetyl group off, and it releases the CoA. The second way that this can happen is by a ping pong mechanism. So the cysteine comes in and attacks acetyl-CoA, and it releases the CoA. And now it's a very high energy intermediate in a catalytic site. And now it binds the second substrate and it coordinates the hydroxyl to be the nucleophile. It attacks the cysteine acetyl group, and now you get acetylation of this hydroxyl. Does any student in the audience know what a ping pong reaction is used for besides acetylation? What if I told you the second substrate might be water? Okay, I'll tell you. It's so cool. It's a protease. Okay, so that's what we thought YAG was. So the cysteine attacks the proteolytic bond, releases the N-terminus, and now you have an acyl enzyme intermediate. The second substrate is water because it's a hydrolysis reaction, and it basically releases the N-terminus of the peptide after it's free. So these two mechanisms are exactly the same. They use the exact same catalytic triad, but one allows water in its active site. The other excludes water. And the acetylation reaction has to exclude water because you can't have something that comes in and hydrolyzes all your acetyl-CoA or you die, because it's a really important intermediate in cells. So what did I tell you about in this first story? So there's a new post-translational modification. There's serine threonine acetylation. No one had heard of that before. That proteases and transferases, they use the same chemistry, but different substrates. And then it also gave us a strategy to study effector proteins or virulence molecules that we don't know what they're doing and we don't know what they're working on. The idea is identify the target, determine how the target is changed. How has it chemically been changed? And then elucidate the biochemical mechanism or the chemistry that's being used for this. And so basically, this is a strategy we've used for a number of effector proteins. It doesn't work for them all because not all of them involve post-translational modifications, but I'm going to tell you briefly another story that we did that followed the same kind of methodology that really broke open a whole new field of science. And so bacteria have clever things that they do. So the next comes, so now we've done the Black Death, Black Pearl, and then now, Black Spot, and now we're at Black Pearl. And the reason that we're there is because when we were working on YAG, there was a homologue of YAG, and it was from this Vibrio species, Vibrio parahemolyticus. And Vibrio parahemolyticus, I'll tell you, is the causal agent for gastroenteritis. And I'll have a slide about that in a minute. But this is another student in my lab that was working on this protein called BOP-S that came from this Vibrio parahemolyticus. So what is this guy? This guy is, Vibrios are really bad. And with global warming, unfortunately, it's not getting any better because mainly they were contained by temperature, because when things got cold, they went into a dormant state and they don't get in your seafood, and it's pretty safe to eat it like in the old days. The old days was 20 years ago. So basically, oysters were okay to eat in any month that had an R in it. So between May and August was not a good time to eat oysters because it was really warm and all the Vibrio are proliferating, right? And they contaminate the raw seafood and then you can get sick. So now it's different because now we've got these heat waves that are coming through, and I'll give you a couple of examples of what's going on. So in British Columbia in 2018, there was a heat wave, and it basically, when they went to harvest the oysters, 80% of their harvest was dead in the bays that had the heat wave. And it's basically because this Vibrio got out and infected the oysters and killed them. In another part of British Columbia, there was an outbreak from eating raw herring eggs, which the indigenous population does as a custom every year. And there was an outbreak of Vibrio cholera in British Columbia. And then the other thing that Vibrios do is they love DNA, and that's a whole nother story with shrimp farms that I can tell you about at another time. So Vibrio parahemolycus, the strain that we work on, causes gastroenteritis due to the consumption of raw contaminated seafood. And I'm going to tell you about 3 virulence factors they have. They have these hemolysins that just make holes in things. They're annoying. And then there's the type 3 secretion system that we consider non-toxic. And that's because it's in every Vibrio parahemolycus strain, including the environmental strains, because there's 2 types. There's clinical isolates that they get from people, and those have this other system, this toxic type 3 secretion system too. But the Vibrios that don't have the system are just basically commensals with shrimp, and they have the first type 3 secretion system 1. So we wanted to study these, and the way that you do this so that you can uncover virulent systems is you have to get rid of one system so it doesn't mask another. So we would take strains and we'd get rid of the hemolysins or get rid of the second system so that we could just focus on this first system. So it's not masked by either one of these. And then if you want to study the second system, you get rid of the first system and the hemolysins so that we can uncover its activity. So the first system, the non-toxic system, is on chromosome 1 in the pathogenicity island. It has about 4 or 5 effectors, and we don't know what it works on, but it's conserved and it's evolutionarily conserved. So it's important for some little animal in the ocean. The second system is a pathogenicity island that's on the 2nd chromosome, and it has its own repertoire of effectors. It's turned on by bile, which I'll tell you about in a minute. And basically it causes this gastroenteritis, but it's important to remember that we are not the host that's causing the evolution of this system. We're incidental hosts. We get sick when we eat raw seafood, but we're not driving the evolution of the system. It's another animal in the ocean. So if we take these 2 strains, the one that only has the first system and the one that has the second system, if we put it into a mouse, this is very weak. It's not a really good pathogen, but you do what you can. All right, so you put it in germ-free mice, and if you put the first system in, it does nothing to the mice. If you put it in the second system, it makes the mice sick. They get a little bit of diarrhea, and they basically start sloughing off the epithelium in their sebum. Now, in contrast to that, if you take the first system and you put it on tissue culture cells, it will kill every type of tissue culture cell within 3 hours with an MOI of 3. So these virulence factors are really, really targeted at very specific things inside the cell. The second system just changed the shape of the cell, which was kind of weird because we thought it would be the reverse. So when I first started working on this system, as I told you, we started working on this guy, VOP-S. Melanie was working on it, and that was involved in the second system. And what it causes is cell rounding. So when it caused cell rounding, we kind of guessed at what it might be working on, and we figured it might be working on G proteins. So Rho GTPases are these G proteins that have an inactive and an active state. When they're active, they're changing the actin cytoskeleton. And this particular one, RAC, causes membrane ruffling, and you can get stress fibers from Rho and different things from CBC42. But in this case, we're looking at RAC. And when it's active, it's found in GTP. And when it's inactive, it's found in GDP. When it's active, it fits like a key into a lock. So it basically can bind to things downstream and then cause changes in the actin cytoskeleton. Now, what we noticed is in the presence of this guy, VOP-S, which is another effector protein from Vibrio, that the Rho GTPases could no longer activate the actin cytoskeleton, and we didn't know why. So what did we do? We decided to incubate VOP-S with RAC and ask what it might be doing, and it changed the molecular weight of the G proteins by 329 Daltons, and that turns out it's equivalent to AMP. So it was modifying these proteins with AMP, and that caused steric hindrance so that they no longer could bind to their downstream effectors. And we called this new post-translational modification, which we thought was new. It actually had been discovered 50 years ago by Earl Statman. I'll tell you about that in a minute, but we called it ampylation, and now the name kind of has stuck. So basically this VOP-S, which contains a FIC domain, can modify things with AMP. Now I want to go back to my earlier slide. So what do? We identified the target, and then we determined how the target is changed, and then we elucidated a biochemical mechanism. And in this case, it was ampylation. So what's ampylation? So you know what phosphorylation is? You take the gamma phosphate with a kinase and you add it to a hydroxyserine 3 tyrosine residue. And then a phosphatase can take that off. Well, ampylation adds an AMP and it uses these PIC domains, that's VOP-S. And then there's a nucleotide transferase domain that Earl Statman had discovered in the 60s. He found one protein that was modified by AMP and they couldn't find any more until we discovered this PIC domain. And ours modified a threonine, but you can also do a serine or a tyrosine. And then it can be taken off by phosphatidyl diesterases, nucleotide transferases, and actually PIC domains can reverse the chemistry as well. And so I won't go in details, but this has been a really cool discovery that we made about 15 years ago. And so basically we discovered this PIC domain that ampylated and other people had PIC domains. Well, this domain had been around for so long that it evolved to use different metabolites. So it not only did ampylation, it does phosphocholination. So it takes the CDP choline and transfers the phosphocholine. So it's the same kind of chemistry. And then umpylation, and then it can flip the ATP in the catalytic site and do phosphorylation. And it turns out that this nucleotide transferase that Earl Statman had studied like 50 years ago has also been hijacked by bacteria for different effector proteins. And then a third enzyme has been linked to ampylation, and it's very cool. So remember I told you about the ampylation and how it looks at the, and the phosphorylation looks at the ATP. Now take a kinase and flip the ATP in the active site. Now it transfers AMP rather than a phosphate. And so all three of these enzymes can carry out this type of biochemistry. And it also can do capping on viruses. So I don't know if anyone's seen this recently, but this is a beautiful study where you're basically, instead of, the chemistry is beautiful and you're basically modifying something with RNA, and it's something that the virus is doing, including COVID. So these domains are used by pathogens. All modifications are inhibitory, and PIC domains are in eukaryotes. So it turns out every metazoan genome has one PIC domain containing protein. So what I told you about was we studied this guy, VOP-S, that was in this system of killing by Vibrio. We solved the structure. We realized it was a PIC domain. Others studied the PIC domains. But then we realized there was a PIC domain in metazoans and one in every genome. And so we studied it in flies. And then we discovered what it worked on in flies. So it's a very conserved mechanism involved in the unfolded protein response. And now we've gone into mice and we found that there's disease models in humans. And now we're making those in mice. So this is the other half of my lab that another person in my lab is actually going to take to use to form her own lab and study these molecules. But it kind of shows you that there's a lot of evolution going on with these bacterial pathogens, and there's a lot of mimicry, as I told you in one of my first slides, that a lot of these mechanisms are used by different systems, and they're regulated in bacteria. Many times the effectors that they use are unregulated. So now what I'm going to do for the last part of my talk is I basically want to tell you a little story about the second system, because this is the part that's closest to my heart. I like the PIC and the ampylation stuff, but that's moved on to others. My heart is really in pathogenesis and host pathogenesis. So what I told you was that this second system is induced by bile acids. And I also told you that when we took this bacteria and we put it on tissue culture cells, we really didn't see anything. We saw all this actin assembly, red is actin, and getting all these actin stress fibers, but we really didn't see anything until, so while we're studying this first system and we're getting all these different virulence activities and having lots of fun with all of these things that are working really well, it was really rather frustrating because we didn't know how this worked. And then I had a really talented postdoc in the lab. And we were so frustrated by not being able to figure out what was going on, except for these changes in actin cytoskeleton. I said, Ling Ling, why don't we go to a meeting together? We'll go to a GTPase meeting. And you can just think about something else and, we're going to figure out, what's going on, but let's just get out of our mind for a while. So we go to this meeting. She used to work on Shigella. And we go to this meeting and she sits back and she goes, Kim, I figured it out. And I said, what did you figure out? She said, I figured out what's happening with Vibrio. And I said, what? She said, well, it's invading. And I go, no, 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 no. I'm a biochemist. I work on extracellular pathogens. I don't work on intracellular pathogens. She goes, well, you do now. And so then 2 weeks later, she went back and did the experiments and she basically showed me what was happening. And so this is a visualization of what happens with Vibrio. It's basically forms a protective replicated niche in epithelial cells. So it goes into, and I'll show you how it does it, but these are really pretty pictures. So it basically divides and divides. It's in the cytoplasm, and then eventually it escapes. And so it's basically, this whole thing happens in about 7 hours. And so this is another, so one of the ways that she figured it out is she basically looked at the type 3 secretion system 2 pathogenicity island, and she said, oh, there's this protein that looks like CNF. It looks like an effector from E. coli, and that facilitates entry into host cells. It looks like a deamidase. And so this is the most conserved post-translational modification you can do, because it changes a protein by 1 Dalton. And basically what it does is it deaminates a glutamine residue to make a glutamate. And so you get a 1 Dalton change, and that constitutively activates RAC. And when RAC is constitutively activated, it causes membrane ruffling. And so basically what Vibrio does is it mediates its own uptake. And this is the surface of a HeLa cell, and purple is my favorite color. So they made Vibrio purple, and what it's doing is causing membrane ruffling so that it can be rolled up into the cell. And then when it goes into the cell, it basically goes in, and BOP-C induces its uptake. It makes a nascent vacuole, and then it makes an endosome. That's an acidification step. And then normally it would fuse with the lysosome and go to the phagolysosome and die, but not Vibrio. What Vibrio does is it escapes into the cytoplasm after the endosome has been acidified. And then it turns off all the host alarms. We can't tell actually which cell is infected and which is not unless we turn on the green filter. And then it escapes. And we know it uses a number of different effector proteins, these BOPs. And we know how it escapes. We discovered the enzyme that allows for its escape. And then we know, I remember I told you about VOP-A, well, it turns on MAP kinase signaling, but it doesn't turn off NF-kappa B, and that makes sense because it doesn't want to kill the cell that it's invaded, right? So it has specificity to these enzymes. And then VOP-L inhibits ROS. Okay, so now what I want to do is I skipped something, sorry. So up there is a little question mark up there. So the question is, how does the whole system get turned on in the very beginning? And I hinted at this in the very beginning because I told you that Vibrio meets up with bile in the gut. And it turns out bile is a really important factor because what it does is Vibrio senses the bile acids, and this turns on its type 3 secretion system. So within about 30 minutes, it turns on the system and builds that needle. And then it basically goes to the epithelium, it injects in these effector proteins, and then it invades the cells. And then they get sloughed off, they make hundreds of themselves in a protective niche, and then they go on to whatever their next host is. But basically what's happened is, in the presence of bile, Vibrio is turned from a friend to a foe. And this is really important because it's going to be dictated by biles. And bile acids are modified by things in the host cell, which I'll tell you in a minute. All right, so now what you're doing is another student in my lab decided he wanted to understand how they were receiving a signal from bile. And there was a bile acid receptor that nobody had studied it well at the molecular level. And what he discovered is there were actually 2 genes that made this beautiful beta barrel that binds to bile acids, and that receives the signal. And it turns out that this receptor is interesting. So you have the outer membrane of the bacteria, the inner membrane of the bacteria. This is the cytosol down here. And it's 2 proteins, and one of the 2 proteins goes through the membrane and it links to a helix-turn helix structure. So that's a transcription factor. And that transcription factor stays linked to the membrane after the bile binds and this receptor is activated. What it does is it turns on a gene in the type 3 secretion system island, and that gene is another transcription factor called VTRB, and it has a helix-turn helix as well. And that, in fact, is the master regulator for the whole type 3 secretion system. And that turns on like 80 genes. And then these 80 genes make all those effector proteins that get injected out of the needle as well as the needle. So it makes this whole structure. And doing so, it has to make chaperones because these guys have got to fold, and it has to make the effectors that get basically ejected out of the cells. So for a long time, I've been looking at this, and I've been trying to convince people. It took me about 6 years that this was interesting, and then my idea was good. And what my idea was is this was all happening on the surface of the membrane of the bacteria, and I think it's all happening in one space. And I think this is a factor. I think that it starts there at one place on the bacteria, and all the stuff is made in this one little space, and it's put into the membrane. Well, then one day somebody came up to me and said, "Ha, your hypothesis has been around since the 70s. And what it's called is the transinsertion hypothesis. And that's actually where you have transcription, translation, and insertion of gene products into this to create a distinct domain. In this case, it was the type 3 secretion system. So finally, KK joins the lab. Oh, wait, he didn't have a poster today. So KK joins the lab, and I convinced KK to work on this because he loves microscopy, and he wants to have, and we got a super res microscope that he could use. So he goes in and he says, okay, I'm going to look at this. And the first thing I'm going to look at is the DNA, because what happens is the DNA has to go from the middle of the cell to the membrane. And so he's going to follow it. And so basically what he did is he labeled the DNA. And the way that he does that is he makes a protein par B with a GFP tag. And then there's a site this binds to, par S. You make a ton of this protein, it binds to the site, and it makes that spot on the DNA green. And so we put that par S site up there in that type 3 secretion system island. So now we look at our cells in the absence of bile, and then we add bile. And what we find out, and this has all been statistically validated by hundreds and hundreds, thousands actually of samples, that basically in the presence of bile, the green dot moves to the membrane. So then the next thing that we wanted, and this is the statistics for it. So then the next thing we wanted to do was we said, does all the DNA move to the membrane? So we labeled the type 3 secretion system with green and the other side of the chromosome with blue. And then we added bile, and what you can see is only the green goes to the membrane, not the blue. So it's particular for that type 3 secretion system, that pathogenicity island. So then we said, well, let's label the transcription factor because it's one of those guys that can float around the membrane because all it's got is a helix in it. And so we label it with superfolded GFP, and it's not there in the presence of bile because it's got to be made, but 30 minutes later it appears and it's right on top of where the island is. So now we've got the DNA and the second transcription factor that's the master regulator for this. And so the next thing we wanted to do was to look at the needle, and the way that this is cool. Okay, so the way we did it is there's no cysteines in the needle. And so we added cysteines where there were serines so that we could use biochemistry to label the needle. And we did that, and we got one cysteine that we knew worked for all the infections that we did. And so in the absence of bile, it's not there. 30 minutes later, it appears. And wherever there's a blue dot, there's always a green dot. But there's not always a blue dot where the other green dots are, partly because this is a confocal. So it's the spike through the image, but also because they're very fragile and they fall off. So then we wanted to make sure the master regulator was where the needle was, and in fact it is. So if you look where the blue, where there's a light blue, there's always a dark blue. And this is actually in the periplasm, right? This is where we mark it in superfolded. And so basically what we think that we have. Oh, and so then we labeled the RNA as well, because if you're making protein, the RNA has to be there. Nobody had ever labeled RNA in bacteria, but KK got it to work. And so we used a phage method for this. And basically where there's RNA, the transcription factor is as well, because that's what it's making. And then finally we did EM. And EM's messy, and it's something you use to support your hypothesis because you can only get so many images. But basically what you can see is that we can fold particles into the needle, the transcription factor, and then we localize to the messy same spot on these cells. So basically what we think is that we have this ligand-induced co-component receptor. I'm going to tell you about that in a second. recruits DNA to the membrane for local transcription, translation, membrane insertion, and assembly of a protein complex, the type 3 secretion system. And so we think it all works at one spot on the DNA. So while we were doing this, we were also looking at bile acids. I don't want to go too long. Were you okay? Good. So this other student in my lab, I'm talking about all my students in my lab. So she decided she wanted to look at bile acids because it turns out not all bile is the same. So if you add bile, you turn on this type 3 secretion system, you make this CTRB. But if you add CDC, you don't turn on the system. If you add TDC, you do turn on the system. She thought, well, are they binding? Are they doing something similar? And she said, because they're really very similar. So if you look at CDC and TDC, and you look at the places where they're modifying, the R1 and R2, and then R3, those are the only differences between these. It's just a flip of a hydroxyl. And the tail that's up there is basically sticking out of the beta barrel that I showed you where the bile acids were binding. So the change of one hydroxyl is changing the way this guy can signal. And so she said, well, are they binding to the same place? And she said, well, they probably are because I can do competitive binding. So if I add the activator and more of the inhibitor, I reduce the signal, and vice versa. The signal's off, I can turn it on with increasing amounts of the TDC, the activator. And the gut microbiome metabolizes bile acids, and that can influence whether you're getting biles that are activating the system or biles that are not. And actually, it does correlate with what you might think in a healthy gut as compared to a diseased gut, but it's very loose correlation. So here's our bile acid receptor. You can see the tail sticking out of the top. So how is that hydroxyl affecting anything? So she wanted to make sure that it was binding, but we also had data from a long time ago from Honda and Aida that TDC can activate and CDC cannot. So then what she decided to do was to do ITC, just look at binding affinities. And what you can see is TDC binds well, CDC binds well, and they're probably binding to the same place. But we didn't have this structure. We had the structure of TDC, but we didn't have this one. So she went and solved the structure with CDC. And what she found was this one little hydroxyl makes a salt bond right there and stabilizes this alpha helix. So the B means stable, red means very flexible. And then if you look at the one with CDC, now this whole thing is very flexible. What I can tell you is that this alpha helix is moving down to where that transcription factor is. So there's a conversation because of that binding across the membrane that's going to basically say be active or don't be active. And then we have this new receptor. So you classically know about one component receptors when you bind a ligand and it produces a transcription factor. Most of those are soluble. I'm showing you there's 10% that are in the membrane. And then there's two components where you have a histidine kinase that's activated and it phosphorylates a reporter from receiver domain to activate the transcription factor. Here we've got all of it happening in one piece. So you've got a basically something binding at the top and it's changing the way the signal is down at the bottom. But we really wanted to find more of these. But the problem was that what I can tell you is the essence of the problem was that we realized after we solved the problem is that this is a very fast evolving protein. And fast evolving proteins say structure, but not sequence. So BOP-C, this guy, this blue guy that makes this beta barrel has no obvious sequence homologies to any other known protein. So how do you find more? So what Lisa did is said, I'm going to take 20 reference genomes, just in bacterial genomes. And then I'm going to say, give me every protein that has a helix turn helix. And then of those proteins, give me all the ones that have a transmembrane sequence. And of those, give me everyone that's in the same operon or adjacent gene that also has another transmembrane sequence, okay? And then the one that's the second one that she found is the operon, then what she can do is she can take AlphaFold and ask what their structure is. And when she did that, she found this. She found all those adjacent genes with a similar structure, but completely diverse sequence. You could never find it by sequence. So essentially what she had was 8 of the 20 reference genomes had co-component receptors. They're all enteric bacteria. they're likely binding a bile component to activate the system. And we know that they're regulators of associated systems. So not necessarily a type 3 secretion system, but maybe a fimbriae or a transporter or another really large component that you probably are going to use transertion to make, and you're responding to the environment to make this kind of thing. So we think that's really cool. And now we've started studying ones in some of your favorite pathogens. And we think that it's just a really cool system. So it's a new virulence factor in a way, it's a new receptor. So what did we learn? Effectors are clever mechanisms to manipulate the host. Microbes are resourceful and use an interplay between virulent systems, housekeeping mechanisms, and host cell resources to evolve ingenious schemes for pathogenicity. What I didn't tell you is one of the mechanisms that Vibrio uses to escape out of the cell is not an effector protein, it's a housekeeping protein. So the pathogenicity island landed in Vibrio, and it was like, it had all this machinery to invade a cell, but then it realized it had another component that could allow it to escape that was a housekeeping gene. And so they really do know the environment that they land in. And in response to environmental cues, complex membrane localized machines can be efficiently assembled for transertion. And always test a hypothesis until you can no longer prove it wrong. And the last thing that I'll say that I don't have up here is bioinformatics can only tell you so much. Until you do the biochemistry and you figure out the mechanism, you really don't know what your enzyme might be doing. So it requires you to do many different things. And so finally, of course, the most important slide is to thank everybody that's worked on all of this and all the collaborations and all the funding supports that we've had. And I just have a great team. It's evolving, it's changing. That's already an old slide, but yeah, that is what labs are. So, and with that, I want to thank you. And then I will go through another little slide, but you want to ask questions first, or what do you want to do?
Next slide, and then we'll.
Okay. How I got here. I told you a little bit about the last 20 years, but we're going to go back even farther. Okay, so I went to Texas A&M, and I'm an Aggie, and I can talk like a Texan. So I got my bachelor's in science in biochemistry. Everything's in biochemistry. So I was at Texas A&M, and then I knew nothing about I was a tech for a year, and then I knew nothing. I didn't know what you did, whether you got a biochemistry degree. They said, oh, you could go to graduate school. So I applied to 2 places, and I ended up at UCLA. And then I left, and I wrote about that. And it was, I didn't take care of myself, because back then you didn't have the internet, say you're supposed to exercise, eat, and sleep well. And so I crashed. And so then I went back to Dallas, and I was a technician in a biochemistry lab where I learned about protein chemistry, which was really important for the virulence factors. We didn't do we did protein sequencing at the time, not mass spec. And then I was in a Drosophila lab, and I learned Drosophila genetics, and I really learned the difference between genetics and biochemistry. They're both valuable, but they tell you very different things. And then, so I asked seminars, I went to seminars and I would ask questions, and this one guy was like, why are you asking questions? You're just a technician. I said, okay, I'll get my membership card. So then I went and I got my PhD, and I was like, okay, now I have my PhD, now I can ask questions. No, but then I didn't really, then I learned a little bit more about science, and I was really lucky, and I got to go to meetings and hear other person's science and everything. And then right after I defended, my husband proposed. And so then I was like, what? And so then he had already was shopping for jobs, because he was a step ahead of me. So he was going to be faculty, and I was going to be a postdoc wherever we ended up going. And then I did a postdoc at University of Michigan, go blue. And then we moved. So all in 1 year, we had new jobs. We bought a house. I got pregnant. And that was like, I didn't know, it's like, really? Okay, so now then we moved up there, and then to Michigan, and then I had 2 kids, and that was amazing. And they're 28 and 25 now. They're little people. They're still little people. And then I did 3.5 postdocs, because I was there, and my husband had his career. And we had kids and everything, and so I just learned different things. I went to different labs, and then we did a job search for 2 people, which was an enlightening thing. And then what happened was UT Southwestern found out we were on the job market because 2 friends had invited us back to give seminars, and my family lived in Dallas. And they said, well, why don't you come back here and work? And I went, I didn't know I could do that. And they said, yes, you can. And I loved it there. It's a great place to work. They have all the toys and everything and really nice people that you can work with. So I'm looking for people. Anybody looking for a postdoc? I've got positions, and other people are there too. I mean, it's just a great place to work. And so then I was an assistant professor, an associate of professor. And then after the 3rd time of applying, I became an HHMI investigator. And then I think now we're empty nesters almost, except my daughter's moving back in, which is wonderful. I love her. So anyways, that's my little trail. So that's it. Oh, yeah.
All right, we have time for a few questions, of course, so who would like to very
Question on the bile acid.
Yep,
Okay, it's really fascinating.
I love it.
Bile acid is like, isn't it different, like you were saying, it's based on the content. So like based on the host, like, it's pretty different.
I was scared about that. Keep
So
going.
people saying earlier that would give you probably involve another host, like do you have any ideas like what that could be, or if it's just based on like, perhaps the type of bile acid?
No, but I made sure those germ-free had CDC and G. I had no idea. I really have no idea what the natural host. I mean, they really like shrimp, but they're killing shrimp a totally different way. It's not using the type 3. It's another story. So yeah, it's a man-made disaster actually. So.
Yeah.
So I was really curious, right? So you said that the gene that is uses to escape the cell is the housekeeping gene. I was curious, how does it escape the vacuum to ensure the cells?
So I don't know. I'm trying to write a grant on that. So what I can tell you, which is weird, is if you disrupt quorum sensing, it can't get out. And I don't know if that's just because you're screwing up all master regulation, because we've tried so many genes to try to figure out how it might be escaping.
It can't get out of the cell, it can't get out of the
It can't it can't get out of the vacuole. The other thing that I can tell you, and this kind of goes to the transertion stuff in a weird way. So if you think about it, Vibrio is only making 1 needle. And so it has to engage that needle, cause membrane ruffling, and then it goes in and it has the needle engaged in the membrane. So it's going to be delivering more things into the cytosol. So we did this experiment because Vibrio is so bad in invading. We thought, oh, we'll get more invasive bacteria because what we'll do is we'll just add CNF, which is a toxin that causes ruffling, and we'll get more Vibrio inside. We got more Vibrio inside, but it couldn't escape. So we think you have to have the needle probably engaged for that as well. And so it's all these little tidbits that you get from different pieces of data that you just have to keep, trying to figure out, but we don't know the answer to that. And the lipase, it's a lipase transferase that basically slowly esterifies cholesterol so that at about 7 hours, the Vibrio through mechanical stress break out of the membrane.
Very interesting topic. So I have 2 questions. So first, when you look at the action of VGLB and making the needle, when you use either fluorescent microscopy or DNA, do you ever see full localization, or is it always on the side of bacteria?
No, it's I didn't answer that right. We do not, we see it about off the pole. It's never at the pole. And it's very interesting that you say that because there's so many other factors that are really, like, I think you know more than I do, just basically on things that can put things out, the SEC pathways and everything. So there's other components that we definitely need to be looking at. There was a professor from Israel that wrote a review on our transertion paper. It just got published like 2 weeks ago or something. And he wrote this beautiful review and he brought up this kind of thing, that there's other factors that you really should consider, especially with exactly what you're saying about where it's not the pole, it's kind of off the pole.
And could you knock out that polarization or the association with the BTRB by deleting maybe the membrane portion? And so they're not.
Okay, so if BTRB is not on the membrane, you don't get activation. Interestingly, if you take VTRA, the first guy, if you get rid of everything in the periplasm and it's in the membrane, it constitutively activates the system. And I think it's because you just need dimerization. We have not looked to see where that is, okay? If you make a soluble transcription factor from VTRA, the first guy, the first transcription factor, the system's dead. And I do think ToxRS is probably doing something. I don't know, you're asking those kinds of questions, yeah. Yeah. Thank you.
So I think you can hear me, right?
Can they hear him on,
Um,
I'm being responsible.
Hi, great talk. I'm wondering if Embryo can also inhibit tyrant chromosomes. I don't believe you showed any cell types in your tissue culture models that would express tyrant.
Right. And it goes into epithelial cells. Do epithelial cells have that?
They do not.
Yeah, that is your answer. Because we've only gone in epithelial cells because we think that's really at least the target for humans, right? And so we basically stuck with Keikos and we're going to do primary epithelial. Yeah, but Keikos, Helos, everybody's Helos are different. So we decided that, yeah. So yeah, no, but there's the gasdermins, the GDPs, all negative. Ubiquin, all negative. So I don't know how it's doing it. So we're trying to make mutants to try to address exactly that kind of thing. But
I found the most last slide.
thank you.
Is there a sense that microphone on that one is not
Is there a question I have to repeat?
Great, well, I think we've had a good conversation and you can always come down and ask any questions if you really want to. Well, let's thank our speaker. Dr. Wolf.
So, no, no. So, oh, good. I hope it was clear. I hadn't given quite that version before. Did it make sense? Most people know about type 3 and stuff. Yeah, yeah.
I think most people know.
Uh-oh. Okay, good.
Looking for first question.
Yeah.